very good afternoon class to everyone, class 12. Good afternoon Aman, good afternoon Neha, good afternoon. So today, good afternoon everyone. So class 12, today we will be starting with the new chapter, that is biotechnology. Okay. Now over here, biotechnology, what do you understand by it? That is, good afternoon Shreyansh, good afternoon everyone. Neha is there, Shreyansh is there, Aman is there. Let's just wait. We will be a bit slow in the starting and then others will also join. Biotech over here means the technology which will be used in the bio. That is biotechnology. Okay. Now this chapter, before that we had completed about the molecular basis of inheritance over here, and what will be the application of it. Okay, application in the sense that why we are studying this topic, biotech. In day-to-day life, if you just see around, the cheese is there, the curd is there, everything what is there, some technology is applied to it. What you are doing in for the curd making, you are just adding the inoculum. Inoculum means a small bit concentration of the bacteria which you are adding in it. Okay, exactly the same form we will also use technology over here in the subject biology. So the application of the biological organism, system or the processes, okay, that will be used over here. Okay, it comprises of genetic engineering. It comprises of genetic engineering. What is engineering? What is engineering? Okay, and second is the aseptic condition. Aseptic conditions. These two are the important, you can say, that are the principles of biotechnology. Okay. So in an applied form, we will be using biotech, we will be using the microbes in the field, and we can get a manipulated genome. Okay, what does it mean by it? You know bacteria? Class 11th में हम लोगों ने बैक्टीरिया पढ़ा था. Bacteria has got two types of chromosome. One is the itself chromosome and another is the extra chromosomal DNA. Extra chromosomal DNA, which we called as plasmid. Okay. Now, suppose I have to multiply. ठीक है, a gene of interest. Gene of interest क्या हुआ? Gene of interest हुआ the gene in which we are interested. Suppose for insulin it is there. ठीक है? Insulin. So a particular group. Good evening, Lakshmi. A particular group of sequences will be there, which we call as genes. And these genes, what is going to do? They are synthesizing insulin. Insulin in day-to-day life, as diabetes is being spread so where, so insulin is being needed. Okay, in the form of humalin. Okay. Good afternoon, Shreya. So this insulin is to be there in the market. लेकिन ये market में है कैसे? ठीक है? Market में means the gene of interest जो insulin secrete कर रही है, उसको हमने multiply किया. Multiply करने के बाद हमें कुछ ऐसे होस्ट ऑर्गेनिज्म उसको मल्टीप्लाई करना था जिससे कि हमें मल्टीपल कॉपीज हमें जल्दी मिल जाए और उसके बाद हम उसको मार्केट में लेकर आए, उससे हमें प्रॉफिट मिले. ठीक है, द बेसिक सेंस अगर हम बात करें एज अ बिजनेसमैन तो हमारा मतलब ये निकल निकलता है. But over here the insulin, we are studying biotechnology. In studying biotechnology, we have to see that how this insulin is being taken out, how it has been inserted. ठीक है. So over here comes the biotechnology. Okay, biotechnology. यूरोपियन फेडरेशन ऑफ बायोटेक्नोलॉजी जो थे उन्होंने एक definition निकाली है बायोटेक की, that is given in your NCERT. That is your first page of your NCERT. You can just go through it. That is the integration of natural science and organism cells, parts thereof and molecular analogues for products and services. It means the integrated science. Now what is that integrated science? This integrated science can be microbiology, okay, biochemistry, molecular biology, genetics. Means these are the different branches of biology which we will be needing in this biotech. ठीक है. Microbiology, it is the study of microbes. Study of microbes means bacteria, viruses, fungi, all which we cannot see with the naked eyes. That is microbes. So the study of microbes is microbiology. Biochemistry, over here we are going to help in it. If we don't know the structure, if we don't know the chemical structure, how we are going to move forward? Next comes the molecular biology. Molecular biology is the study of the genome, okay, of the genes, and we will going to use the molecular tools in it. Okay, genetics is the study of the genes of the genomes that we have studied in the chapter molecular biology, molecular basis of inheritance. Okay, molecular basis of inheritance. Now genetic engineering and the aseptic condition, these two are very important. Now I just show you a flow chart. मैं आपको एक flow chart show करती हूँ, ठीक है. Now what comes the principles in genetic engineering? Sexual reproduction. Only sexual reproduction will lead to variation. And this variation is occurring because of crossing over at the packaging stage of the meiosis 1. Okay. Now this variation which is occurring, okay, which is not possible in the asexual reproduction. This variation can lead to hybridization. Either it can't lead to hybridization also. Okay, now in hybridization we can get a desired gene or we can get a desired gene of interest. Either we will not be able to get the desired gene of interest. Okay, as what is happening in both, multiplication is there. Okay, fine. Now we will use this process of hybridization. That is the mixing up. Okay, so genetic engineering involves manipulation. क्या होता है? ठीक है, मैनिपुलेशन होता है जब हमारा कोई भी डेटा जो होता है कभी कभी रॉन्ग हो जाता है. ठीक है, तो हम क्या करते हैं, उसमें एक जीन ऑफ इंटरेस्ट चाहिए. उसके लिए हम क्या कर रहे हैं, एक होस्ट ऑर्गेनिज्म ले रहे हैं और उसमें हम मैनिपुलेशन कर रहे हैं सो दैट वी कैन गेट द मल्टीपल कॉपीज ऑफ द जीन ऑफ द इंटरेस्ट, एंड दैट एग्जैक्टली इज जेनेटिक इंजीनियरिंग. ठीक है, genetic engineering means the engineering science which will be applicable in the genes. Okay, So it involves manipulation of the genetic materials towards the desired end in a predetermined way. Predetermined way का मतलब हुआ जहां पर पूरी हमारी प्लानिंग होगी, ठीक है. हम हिट एंड ट्रायल मेथड नहीं करेंगे. We will have the full what is the desired gene and then we will move forward. That is why that is in the predetermined way. Okay. Now whatever I have said, whatever I have spoken till yet about the genetic engineering, for that a word will come, that is RDT, that is recombinant DNA technology. ठीक है. ये क्या होता है? Just see. हाइब्रिडाइजेशन में क्या होता है? हमने आर्टिफिशियल हाइब्रिडाइजेशन पढ़ा है ना. क्रॉस पॉलिनेशन कराएंगे. क्रॉस पॉलिनेशन ऑफ द स्टिग्मा एंड द एंथर. ठीक है. यहाँ पे हाइब्रिडाइजेशन हम पर्टिकुलर जीन का हाइब्रिडाइजेशन. What is recombinant? What is recombinant DNA technology? That is related to this genetic engineering only. ठीक है. Recombinant means recombine. जैसे ये है gene and over here we are going to add another gene, a piece of gene of interest. So what it will become? This will be RDT, that is the recombinant DNA technology. Okay. And when it is going to multiply, we will say that it is going to make the clone, and the process will be called as cloning. The process will be called as cloning. Okay. Now, what do engineers do? Engineers design, they construct, they manipulate. Okay. They design, they construct and they manipulate. Genetic engineering will be the same, where we are going to design, where we are going to construct, we are going to manipulate in the genes. That is what is genetic engineering. Is this clear to everyone? Shreya, Neha, Aman, Shreyansh, is this clear to everyone? Please do revert back that you are getting the things. Please do revert back. Okay. Now, what we will be needing? What we will be needing for it? What all are the requirements for it? We will now see to it. Okay, Neha, Shreyansh. What about the rest? Aman. We will be needing the set of enzymes which are very much necessary. Then we will be needing the allied organism. Allied organism means this is the foreign organism. Okay. Then the origin of replication. Okay. And then we will be needing the host cell. अब आप कहोगे मैम ने तो बहुत बड़ी बात कर दी इसमें. ठीक है. इससे पहले हम बेस देख लेते हैं कि एलाइन ऑर्गेनिज्म क्या होता है, ये सेट ऑफ ऑर्गेनिज्म क्या होता है, ओरिजिन ऑफ रेप्लिकेशन क्या है, होस्ट सेल. देयर आर टू बैक्टीरिया, ए बैक्टीरिया एंड बी बैक्टीरिया इज देयर. ठीक है. और एक I'm saying that this is antibiotic resistance and B is not. ठीक है. Now by using the genetic engineering in the aseptic का मतलब होता है that is contamination free. Aseptic means that the condition where it will be be contamination free है, that is being shown because of its genetic makeup. अगर अब ये जो B है, ठीक है, ये एंटीबायोटिक रेजिस्टेंट नहीं है, इसको हमें एंटीबायोटिक रेजिस्टेंट बनाना है. This particular group of genes, which is making it in this B bacteria, then this will also become antibiotic resistant. ये भी antibiotic resistant बन जाएगा, नहीं बन जाएगा? ठीक है? This is what is genetic engineering. Okay? This is what is genetic engineering. But यहाँ पे तो हमने एक बैक्टीरिया और दूसरा बैक्टीरिया लिया है. But get the multiple copies. Okay? This is what is biotechnology. एक और एग्जाम्पल लेते हैं. This was for antibiotic resistant. एक और एग्जाम्पल लेते हैं. ठीक है. Suppose in the DNA fingerprinting की बात है. If suppose we have a small piece of gene, ठीक है, and we have to work on it. We have to go for research of it. ठीक है. We need the multiple copies of it. और उसकी हमें multiple copies कैसे मिलेंगी? We are going to take the host, that is the bacteria. That is the bacteria. और बैक्टीरिया हम इसलिए लेंगे क्योंकि इसके पास होता है प्लाज्मिड, which has got the capability to take in genes, the nucleotide sequences, and with it is going to multiply. That is extra chromosomal DNA. So that particular gene of interest is going to get inserted in that bacteria. Okay, in the plasmid, it is going to get inserted over here, and when this bacteria is going to multiply, it is going to multiply with this bacteria. My gene of interest will also multiply, and when it is going to multiply, I will get multiple copies and I can extract that gene of interest. ठीक है? And I will get the more amount of that particular gene of interest. लेकिन ये कहानी जितनी सुनने में इतनी इजी लग रही है कि हाँ हमने एक जीन ऑफ इंटरेस्ट लिया, इंसर्ट किया, उसको निकाला, ये इतना इजी वर्क नहीं है. ओके, अ ब्रांच ऑफ साइंस, बायोटेक्नोलॉजी, जेनेटिक इंजीनियरिंग एंड द एसेप्टिक कंडीशंस, ठीक है, ऑल दीज आर देयर, दैट शुड बी देयर. ठीक है, then only it is possible. And many of time I have told you कि कुछ भी जादू से नहीं होता है. ठीक है, everything needs enzymes की जरूरत भी होती है. ठीक है, so we have to provide all that atmosphere. हमें वो temperature provide करना होगा, हमें वो enzymes provide करने होंगे so that it can multiply. Okay, so that is what is the aseptic conditions and this is what is genetic engineering. ठीक है, अभी जो मैंने आपको एग्जाम्पल्स बताए ये सारे लेमैन लैंग्वेज के एग्जाम्पल्स थे. ठीक है, इन अ नॉन बायोलॉजिकल टर्म कि ऐसा होता है, लेकिन व्हाट आर द प्रॉपर कंडीशंस. ठीक है, व्हाट इज जेनेटिक इंजीनियरिंग, व्हाट इज दिस alien organism, what is the host body, what are the group of enzymes, the restriction enzymes, the molecular scissors that we are going to deal with. That is what is biotechnology. I hope so that this concept must be clear to you. That is desirable genes. Okay. Now, desirable gene or gene of interest किसे कहते हैं? Many times I am just speaking out that bacteria E. coli, Escherichia coli beta, bacteria E. coli, what you want to clear? Aman, please do tell me what you want to clear in this. Please do specify. Okay students, one thing more I want to tell you that the gene of interest or the desirable gene का मतलब क्या होता है. ठीक है, that is gene of interest का मतलब क्या होता है. जीन ऑफ इंटरेस्ट का मतलब होता है कि जैसे कि वी नीड इंसुलिन. ठीक है, सो दोस पर्टिकुलर जीन विच इज सिंथेसाइजिंग इंसुलिन will be the gene of interest, that will be the desirable gene. ठीक है, thyroid is also there. Okay, any gene which we will be needing it for multiplication. E. coli bacteria used to multiply. Any bacteria can be used to that can be Staphylococcus aureus also. It can be Typhimurium also. I just gave you the example that E. coli जैसे कि इसे 20 minutes में multiply करता है. I just told you in this manner. Okay. So the gene of interest is that the particular group or particular gene for which we will be needing it, on which we will be working on, that is the desirable gene, that is the gene of interest. I hope so that this thing is clear to everyone. To the three organisms.